بس باقي this one's not entered. لازم يسوي F5 عشان
function and this function.
okay, it's supposed to come now. Welcome again. I know it's the end of the second day and everyone is tired, so I'll just try to make it a very short and brief presentation. Frontiers in Autoimmune Research. Outlines for this project, for this presentation will be, I'll be talking about T follicular helper cells, the role of T follicular helper cells with SLE and rheumatoid arthritis, and a couple of slides on peptide immunotherapy and some immune checkpoints as therapeutic targets. The traditional concept that effector T helper cells responses are mainly mediated by T helper 1 and T helper 2 cell subtypes has changed by the recent demonstration of two new effector T helper cells, which is the T helper 17 and a new subset called T follicular helper cells. The T follicular helper cells are CD4 T cells found in humans and have been identified as a new T helper subset. These cells are found within secondary lymphoid tissues and organs, and they are specialized to regulate the development of effector and memory B cells and long-lived plasma cells. It has been noticed and documented that the interaction between T follicular helper cells and B cells leads to activation of B cells and germinal cell formation. It is well documented now that these T follicular helper cells are critical for effectiveness of antibody responses, and as you can see from the picture here that there is lots of co-molecules between T follicular helper cells and B cells, leading to the production of antibodies through memory B cells and plasma B cells. The identification of these cells using flow cytometry, it has been shown that it has a distinct subtype due to its characteristic cell surface markers and cytokine profile. So these cells are rich in CXCR5, ICOS, PD1, and the transcript factor BCL6. And also they are heavily producing IL-21. It has been shown that T follicular helper cells numbers strongly correlate with the numbers of germinal center B cells. And this is just a figure showing that from a previous study that the numbers of T follicular helper cells strongly correlate with the number of germinal center B cells. Now I'll be talking about some published data on T follicular helper cells and autoimmunity. There have been lots of emerging evidence that implicates that T follicular helper cells and the development of numerous human pathologies, including immunodeficiency, lymphoma, and autoimmunity. Uncontrolled T follicular helper cells develop development activates autoreactive B cells to produce autoantibodies, leading to the development of several autoimmune diseases. Recently, it has been published that a significant increasement of TFH has been reported in patients with various autoimmune diseases, including SLE, rheumatoid arthritis, Sjogren, and other autoimmune disorders. Higher frequency of these cells was positively correlated with the serum autoantibodies in these autoimmune patients. There have been several studies showing that SLE autoantibodies has strongly been correlated with T follicular helper cells, and they have been also showed that these autoantibodies are involved in tissue damage. It has been shown previously that indicate that indirect role of T follicular cells in promoting human autoimmunity has been documented by several studies before T follicular was identified. So there was studies showing that production of pathogenic autoantibodies in SLE patient caused by dysregulated germinal center reaction, the presence of the aggregates of T and B cells containing plasma blasts in the kidneys of patients with lupus nephritis also reduce germinal center reaction upon blocking the CD40 interaction in patients with active SLE. There was also recently studies further provided clearer evidence on the role of T follicular helper cells, promotion of both systemic and organ-specific autoimmunity. There was a report showing that circulating T follicular helper cells was increased in SLE and Sjogren syndrome patients as compared to healthy controls. This increasement also was correlated with a clear increasement in anti-double strand antibodies, and an end organ damage in SLE patient was strongly correlated with the number of circulating T follicular helper cells. There was also lots of other studies showing that other autoimmune antibodies such as SSA was strongly correlated with the disease activity. So this study was clearly showing that whenever there is a high percentage of T follicular helper cells in SLE patient compared to normal healthy people, and also there was a strong correlation with other factors. CXCL13, which is a chemokine involved in the T follicular helper cells and B cell trafficking for activation for autoreactive T cells and autoantibody production during the development of lupus, plasma CXCL13 and IL-21 levels were significantly higher in SLE patient compared to normal people. These CXCL13 levels increasement significantly correlate with the systemic lupus erythematosus disease activity index score in SLE patients. Inappropriate immune responses to a self-antigen triggers a pathogenic T cell responses, resulting in a damage to a specific tissue and organs. As I mentioned, that it has been used for a while for other conditions, and this can be achieved by the expansion of regulatory T cell pool and the deletion and/or energy of the pathogenic T cell population. Peptide immunotherapy has been clinically studied in type 1 diabetes and showing initial promising results. Other studies also have been showing that modulation of the antigenic specific T cell responses from predominantly interferon gamma response to a more T helper 2 bias was detected. Clinical studies also have focused on either a single peptide to modulate the pathogenic T cell response or a multiple peptide from the same antigen. And also it has the same method has been used, either single or multiple, for these type of condition, type 1 diabetes. Immune checkpoints as therapeutic targets, I will not go in deep details. Fayhan, mashallah, gave a lot of insight in these things. But just to show you that targeting this type of therapy it might be very successful. PD1, CTL4 are very important checkpoints and might be a very promising immune therapy, especially in autoimmunity. As mentioned earlier, that the association of immune checkpoints receptors with autoimmunity is seen when these receptors are blocked therapeutically in autoimmune patients, and these evidence offer a crucial role played by these pathways in regulating the immune responses. And it also arises the possibility that inducting signals through these receptors could switch off these immune responses and drive the immune system back towards a state of tolerance after control has lost in autoimmunity. These are other studies showing the same, that using these checkpoints are promising for autoimmune disorders. And just to summarize what I have been saying, that T follicular helper cells have been identified as a new T helper subset, specialized to regulate the development of effector and memory B cells and long-lived plasma cells. There is emerging evidence that implicates that these T follicular cells and the development of autoimmunity, and peptide immunotherapy offers a potential strategy to prevent tissue destruction and in autoimmunity. And in immune checkpoints as therapeutic targets showed promising finding in autoimmune patients. Thank you very much.
Thank you, Dr. Abdullah, for this nice presentation. So we reached now